Hi, my name is Phillip Baker. I am a biochemistry major, and I worked with Dr. Bidisha Sengupta on analyzing chlorinated wastewater and its potency to form microbial biofilm. Biofilm is formed when typically single-celled bacteria attach to a surface and aggregate, attach to each other, and act as a multicellular organism. They do this in order to gain environmental resistance, antibiotic resistance, and better allocate resources amongst themselves. Biofilm is important to study because of the antibiotic resistance they have. They pose a significant risk to public health because if you get infected with biofilm, they are extremely difficult to treat by typical methods. The goal of this project was to use microscopic, spectroscopic, and chromatographic techniques to determine the physical and chemical properties of biofilm in wastewater, chlorinated wastewater. Chlorinated wastewater samples were collected from 3 different locations, that being Nacogdoches, San Jacinto, and San Agustin wastewater treatment plants. The 3 samples were put into a 24-well plate alongside sterilized water and tap water, and each well contained 50% LB and 50% sample, as well as however many microliters were calculated to give each sample an optical density of 0.03. Bright field microscopy was used to quantify the amount of biofilm growth. We found that lighter spots in the image indicated vertical growth of the biofilm. Scanning electron microscopy was also used in order to quantify the amount of biofilm growth by literally measuring the thickness of the biofilm, as well as measuring the height of various aggregations on top of the existing biofilm to get an actual measurement. Ion chromatography was also used to provide the chemical properties of the biofilm. It gave us specific ion concentrations in the supernatant of the biofilm samples. We use that data to establish relationships between specific ions and biofilm growth. UV-vis spectroscopy was also used as a way to determine initially if any biofilm growth had happened at all. We measured the supernatant of the sample to determine whether or not biofilm had actually grown. And some conclusions we drew were that Nacogdoches wastewater treatment plant, the chlorinated wastewater at Nacogdoches treatment plant, had the highest potency to form microbial biofilm. We also found that bacteria excreted chloride at a rate inversely proportional to biofilm growth. We also found that phosphate is used by biofilm in order to strengthen the bonds between each bacteria to strengthen the overall stability of the biofilm. And I would like to thank the S.U.R.E program for providing me the opportunity to do this research.